Okay, we're ready to begin chapter 1 in the microbiology text. So let me begin by doing a screen share and pulling up the PowerPoint, which you can find in the Blackboard course shell in the folder entitled Chapter PowerPoints. So as we look at each of these chapter PowerPoints, you're going to notice that every chapter starts with a case study. And so while we may or may not make any comment about that during lecture or during the Zoom class that I'm recording right now, I do want you to read those case studies. They're very pertinent in terms of having information that either directly or peripherally relates to the topic that the chapter covers. Plus, they're just really, they're interesting. So do spend some time looking over that section. It's generally rather short, but it's possible I could ask you a question on these introductory case studies. So please make sure you look at those. Okay. So chapter 1, like most chapters in most textbooks, is an introductory chapter that sort of provides a broad overview of the major themes of the text. And of course, here we're talking about microbiology, so we will indeed talk about some major themes. I'm going to actually divide the first chapter into 2 Zoom presentations. We'll do about half of the chapter in this particular Zoom, and then I'll follow that up with the second half of chapter 1, both of which again will be posted in the course shell in Blackboard. So when we look at the word microbiology and kind of dissect that term, biology is the study of life, and the prefix micro, of course, refers to small. Microscope, right? Using this device to look at very, very tiny objects. In fact, we're going to be doing that very shortly in lab. For those of you that are taking lab, that is, but you've all used a microscope, so you know that word, you know what that instrument does. So the field of microbiology is generally, for the most part, involved in looking at very, very small objects. In fact, up until the advent of the microscope and invention of the microscope, this whole broad field of biology was really unknown, wasn't it? Because we couldn't see bacteria. We can't see viruses with the naked eye. We often talk about these microbes, like bacteria and viruses, for example, as being ubiquitous. Anybody know what that word means? Maybe you've heard of the term. It's certainly not used in our everyday language. Air is ubiquitous. In other words, it's everywhere. And the same can be applied to microbes in our world, whether it's in the water we drink, or the food we eat, or the air we breathe even. Microbes are everywhere, and this will be very noticeable when we start to work with them, especially in the lab. In fact, before we even start working in the laboratory, we have to wipe our tabletops down to ensure that there are no undesirable microbes that could possibly contaminate our cultures. So they're everywhere. And they're not always bad. I just want to make sure from the get-go that we underscore this notion that not all bacteria, not all viruses are out to kill us, all right? Just because we've been hearing a lot about COVID-19 the last year now almost, that doesn't mean that there aren't good beneficial microbes. And we're going to be talking about both pathogenic types and friendly, if you will, types coming up in the next few chapters. So the first few pages of chapter 1 provide you with an overview of what a microbiologist does, or what subfields of microbiology exist. And it's a very broad array of different fields. And I'm not going to go into each of these. I think you can certainly just read over these different sections, especially in these different tables that talk about what public health microbiology might involve. Of course, again, that's in the news constantly. Not that many years ago, Zika, the Zika virus, was of major concern. So that's what this little box on the left-hand side of slide number 5 talks a little bit about. Many pregnant women were very worried about being bitten by the mosquito that carries the virus. We call this a vector. The vector is often an insect, can be other animals too, but mosquitoes often vector different kinds of viruses, or they can vector different kinds of protozoa that can cause you to get malaria, for example. The tsetse fly, you've maybe heard of that. That vectors African sleeping sickness. We'll be talking in an upcoming chapter about different sorts of vectors. But this particular mosquito, whose genus I forget, if that was carrying the Zika virus and it bit a woman who was pregnant, and you may or may not know that it's always the female mosquitoes that bite. Male mosquitoes don't do that. The female mosquitoes need the protein in our bloodstream. So here's, of course, a mosquito injecting the host, if you will, with the Zika virus, potentially, if it carries it. And it's also absorbing, sucking up the blood, too, for the protein that it needs to provide its eggs with. And so what they discovered, about 6 or 7 years ago when this was in the news, was that if you, again, were pregnant and you had exposure to the Zika virus, there was an increased risk of microcephaly in your child. And we see a couple children here who are exhibiting microcephaly, this brain disorder where the brain does not fully form the way it's supposed to. And along with this comes different cognitive deficits and physical deficits. It's a very sad thing to see a child born with microcephaly. This is not the only way you can get it, but it's one of the major causes of microcephaly is the acquisition of different kinds of viruses like Zika. So back to this notion of a broad array of fields and occupations that microbiologists find themselves within. So just take some time, again, check it out, get a sense of the diversity, and we'll talk a little bit more about a couple examples in just a few moments. We will also be talking in an upcoming chapter about the difference between prokaryotic cells and eukaryotic cells. And I think you've all heard of those two words. Remember, prokaryotic cells are much more primitive types of cells. In fact, if you look at the word prokaryotic and dissect its origin, its word origin, pro means before, karyon means kernel, so before kernel. What this gets at is the idea that these cells evolved earliest in Earth's history. In fact, we can go back in the fossil record and find evidence of prokaryotic cells that existed on Earth 4 billion years ago. And some of these descendants of those early prokaryotes are still around today, which is sort of interesting. So they've been on Earth for a heck of a long time. And this sort of schematic here at the bottom of the slide, I think does a really nice job giving us a sense of a timetable. Okay, so here is the origin of the Earth as best we can determine, some 4.75 to 5 billion years ago, with the Big Bang taking place some 14 or 15 plus billion years ago for that. And here we see the formation or evolution of the earliest prokaryotes, again, somewhere between 3.5 to 4 billion years ago. And we'll talk about some possible hypotheses as to why scientists think, or how scientists think, these first early prokaryotes came to form, if you will, or Some would argue maybe they came from other worlds. Interesting hypothesis. But we know that they were here 3.5 to 4 billion years ago. Way, 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 way before you as a human appeared. So here's Homo sapiens. You know, we were here a very, very short time frame compared to prokaryotes. Shortly after, well, shortly being a relative term, because it wasn't really very short a time, but here we see the evolution of the eukaryotic cells. EU means true, karyon again means kernel. By now, I hope you know what the kernel term is referring to. This is referring to the nucleus, okay, because eukaryotic cells have a membrane-bound nucleus. Prokaryotes don't, right? And so these earliest eukaryotic cells have been found in the fossil record to date back some 1.75 billion years ago or so. And we feel, evidence seems to indicate quite strongly, that the eukaryotic cell type evolved from the prokaryotic cell type. And again, we will discuss that in an upcoming chapter. But what I want you to, again, note is that for many, many, many billions of years, right, almost 2 or more, the prokaryotes ruled supreme on Earth. They were the only type of cell found. With animals, reptiles, and insects, and mammals evolving much, much later in Earth's history. Okay, so I've talked a little bit about the difference between prokaryotic and eukaryotic cells. You should have already known this from other biology courses that you've taken, perhaps. And we will spend an entire chapter on the prokaryotic cells and another chapter on the eukaryotic cells as it relates to microbes. So there's going to be a lot more coming up on this. But this set of diagrams at the bottom of the slide, again, sort of reiterates the notion that prokaryotes don't have membrane-bound nuclei, and eukaryotes do. In addition, eukaryotic cells have a lot of other additional membranous organelles. You've heard of ribosomes. You've heard of smooth and rough endoplasmic reticulum, right? You've heard of lysosomes. You've heard of Golgi bodies. Those are all membranous organelles that have specific important functions within the eukaryotic cell. That doesn't mean that prokaryotes don't have DNA or chromosomes, which we typically associate with the nucleus. They do. Here you see a chromosome in a bacterial cell. This could be a bacterial cell, a prokaryote. They do happen to have ribosomes, which is really the only organelle, for the most part, that they share with eukaryotes. And you might remember from A&P or your biology classes in the past, ribosomes are the sites for protein production. It's where proteins get assembled. Very important process. So more on a review of prokaryotes and eukaryotes coming up. Actually, starting next week, I think we do prokaryotes. Another example of a microbe that I know you've all heard of, and I've alluded to this just in one of the earlier slides, and that is the group of microbes called viruses. Now, what's really interesting about viruses, which we devoted an entire chapter to as well coming up, is the fact that these are acellular. What does that mean? The prefix a means without. So these are not cells. They're very, very different from eukaryotic cells or prokaryotic cells. They are non-living. They require a host cell to inject their nucleic acid into. That could be DNA or RNA. And then what the host cell does is it utilizes that genetic information and it assembles more nucleic acid, and it makes proteins from the instruction booklet provided by the virus. It makes more viral proteins and more viral nucleic acids. And basically what then results is the host cell that's been invaded will assemble new viral particles within it and release those out into the environment, where they in turn can invade additional host cells with their nucleic acid, and those host cells are turned into viral factories, basically, which eventually leave the cell and go on to invade more and more and more host cells. So again, more information coming up on viruses in an upcoming chapter that we will talk a lot about different kinds of viruses and how they work. But in this particular diagram, you see a couple sketches. This is the HIV virus, which causes AIDS. And this is a bacteriophage, which is a virus that targets bacteria. But note that both of them contain a nucleic acid core, illustrated here in the red. So this is going to be either DNA or RNA. In the case of the bacteriophage, it's DNA. In the case of the HIV virus, it's an RNA virus. And then we have an outer protein coat, illustrated here in blue. Now, note that this particular virus has an outer envelope. This guy does not. And we'll talk more about this again in an upcoming chapter. So we call this an envelope virus. This would be called a naked virus because it doesn't have an envelope. We're going to also be expanding our thinking when it comes to microbes as not including simply or solely the bacteria and the viruses. And this slide kind of talks a little bit about some additional forms of microbes that we can and will talk about. So in addition to bacteria and viruses, which you see on the left-hand column here of this page, are some additional photographs of some other microorganisms, which include fungi and protozoa, algae, and helminth worms, they're called. Now, when we talk about these four categories or groups of microbes, the fungi, the algae, the protozoa, and the helminth worms, these are all composed of eukaryotic cells. They may be unicellular, like we see here in this micrograph of a ciliate, or in the case of certain algae, they might be unicellular. Even some fungi can be made up of one cell. But there are examples of these four groups that are also multicellular too. Although most protozoa, I think, are unicellular. But you can have multicellular fungi, multicellular algae, and multicellular worms. In other words, made up of more than one cell. So we will be talking more about this group of microbes in upcoming chapters. But again, expand your thinking now. Don't think of just bacteria and viruses, even though that's what we generally focus more on, but there are other types of microbes too. Whether we talk about viruses or bacteria or protozoa or fungi or whatever the case may be, we want to get some sense of the size. And I really like this figure in your book that takes you from very large, relatively large structures all the way down to very, very small structures, down to the atomic level here. So we have here in sort of dark pink, the atomic level. As we work our way up, here we've got things that we can see, but only using an electron microscope. And then as we get into larger structures, we can use our compound light microscope in the lab to see these guys. And then we can use our naked eye to see other types of structures. So when we talk about the size of microbes, keeping in mind again that we're not talking just about bacteria or viruses, but it could be a roundworm, or it could be a reproductive structure of a fungus. We have to be a little knowledgeable about different sorts of units that scientists use to describe how large a microbe is. Now, you have all worked with meter sticks. In fact, in A&P1, we do a lab where we use a scientific method to explore the relationship between long bone length and height. You might remember doing our lab report on that. So you've all hopefully worked with a meter stick. And that's what's, of course, photographed here, a little sketch of it. And you remember, I hope, that this meter is made up of 100 centimeters or 1,000 millimeters, right? 1 millimeter is 1/1000th of a meter. So if you look at a meter stick, you look at all those little, small little increments, the smallest shown on the meter stick, each one of those is a millimeter. And there are 10 of those per centimeter. There are 100 centimeters. So 100 times 10 is 1,000. So what I've done now is I've taken a closer look at a small piece of that meter stick, the first 6 centimeters here, basically this end of the meter stick, and I've blown that up. And here we can see the 1, 2, 3, 4, 5, 6. These are centimeters. You might say, well, why aren't they millimeters? There's mm. mm are these individual small units, as I mentioned a moment ago, that you can see with the naked eye if you look at a meter stick. And there are 10 of those in each centimeter. Okay, so how many millimeters do we have in this photograph of this part of the meter stick? Well, we have 6 centimeters. We have 10 per centimeter. So the answer is 60. There are 60 millimeters on this piece of meter stick. All right, now I want you to take that millimeter that you can see here, this small little unit, and I want you to divide that 1,000 times. Take that millimeter, subdivide it 1,000 times. If you do that, each one of those little subdivisions would be called a micrometer. Micrometer, and we use this kind of funny symbol to indicate micrometer. Looks sort of like a U, but it's not. It's a micrometer unit. And so if we look at 1 micrometer, it is 1/1000th of a millimeter, right? Because remember, I want you to take one of those little millimeters, divide it up 1,000 times. Each one of those is a micrometer. So there are 1,000 micrometers in a millimeter. And the reason that I bring up this notion of the micrometer is because when you start to look at some of these units that define different size microbes, there's that funny that's micrometer. Okay. So when we look at protozoa, which we will in lab very soon, or mold spores, or certain types of bacteria, like we see here, we're going to be using micrometers to indicate their sizes. We can't see them with the naked eye. We would have to use a compound light microscope. Now, let's expand our thinking just a little bit more. Let's take one of those micrometers, one of those 1,000 units, right, within each millimeter, and let's divide the micrometer up 1,000 times. If we did that, we would have a nanometer, or nm, nanometer. So we're getting down into some very, 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 very small sizes, aren't we? Units to describe lengths. And as we get into the range of the electron microscope, and down into the atomic level, i.e. the distance between the nucleus and the first orbital of an atom, notice we're moving now from micrometer to nm, nanometer, to describe the size of different kinds of viruses. Here's HIV we talked about a few moments ago. By the way, the coronavirus would be down in this category too, right? We'd use nanometers to describe the size of the coronavirus. And then, of course, as we get even smaller than viruses, we're getting into now actually macromolecules like DNA, which is a nucleic acid, double helix. Here's protein, made up of amino acids, right? Lipids, carbohydrates, nanometer to describe them. So again, I think it's just helpful to have some appreciation of microbial dimensions and sizes. Microbes have a real important, unique role to play in our world. And I'm going to be shifting again away from the pathological viewpoint for just a few moments and thinking about them from an ecological point of view. Now, in high school, you remember studying photosynthesis, I would hope. This ability of plants and other organisms, usually they're green in color because they have chlorophyll in their cellular structure, in the chloroplasts. These organisms are able to take the energy from the sun and convert it into usable energy for the cell. And what is often done is that plant takes gaseous carbon dioxide and converts it into oxygen, which we can breathe, right? And we do breathe. So we rely on photosynthetic organisms all the time, plants in particular. But what's quite interesting, and I think a lot of people don't really appreciate or realize this, is that most of the oxygen that we breathe, that you're breathing as we speak, that you listen to this presentation, most of it comes not from trees, but rather from planktonic organisms like algae. Okay, so in the rivers and lakes and streams and the oceans of our world live these phytoplankton. And they, like plants, are photosynthetic, and they're utilizing the sun's energy and they're converting the CO2 into oxygen. And that's what's shown, of course, up here in the upper corner of the slide. We also, if we stop and think about microbes, should appreciate the role they play in nutrient recycling and in decomposition. So what once was, for example, a very barren landscape on early Earth billions of years ago can and was slowly transformed into some primitive soils by bacteria and other microbes that lived basically on the rocks. And they broke that granite down and that rock down, and certain airborne particles got caught in that debris field, if you will, and over many, many millions of years, soils develop. But it's really the breakdown of that early Earth, if you will, that set the stage for the acquisition and development of sediment, which in turn provided a substrate for other microbes to grow. We don't really think too much about that. We certainly can understand this photograph because we've all gone into our refrigerator and or in the crisper of our refrigerator and found that package of strawberries or that orange or that grapefruit or whatever it might have been that we kind of forgot about. We pull it out and it's covered with some beautiful fungi or maybe even bacterial growth, right? It's gone bad, spoiled. And because microbes like fungi are ubiquitous, they're in the refrigerator, they're in the air on your countertop. It's only a matter of time before they begin to utilize the energy stores in that fruit, in that vegetable, or whatever it might be. And they are extremely important in decomposition, breaking down organic materials, right? When a tree falls over in the forest, or when an animal dies in the forest, it eventually is decomposed by fungi and other microorganisms. And those molecules that made up that tree or made up that animal eventually are recycled into the ecosystem, mostly by microbes. So they're extremely important. We make use of microorganisms. We've been able to manipulate them for the last 50 plus years. And that's what this slide kind of gets at. We'll be talking more about this, and you'll have read over some of this in chapter 1 as well. But this subfield of biology called biotechnology is a very broad field. And I think I would probably even put genetic engineering and bioremediation underneath biotechnology, even though we list them as 3 separate entities. These sort of ladder, I think, from biotechnology. So the manipulation of microorganisms to allow us to make products that improve our lives, whether that means to form vaccines. And all we have to do is look at many of the current coronavirus vaccines. And I chose obviously here Pfizer and Moderna because they've been in the news a lot lately, but there are other additional vaccines that are being developed and I'm sure will be rolled out publicly in the next few months. These make use of messenger RNA. I'm not going to get into the details of that right now, but it was a result of genetically manipulating the current coronavirus, manipulating the genes of that virus to develop a vaccine that we are then injected with, and our immune system responds to that messenger RNA vaccine of the virus to form antibodies and to build up a memory to those particular spikes on the surface of the coronavirus, so that if we end up getting it via our neighbor, if you will, or down at the grocery store, our immune system will be able to recognize those spikes on the surface of the virus, and we won't get infected with it. Bioremediation is the use of basically microbes in helping to clean up oil spills, to degrade poop. That's what septic tanks are basically for, right? They degrade undigestible organic compounds in a safe way. So we make use and have made use of bacteria and many of these microbes for a long time. The genetic engineering of the biotechnology is much more recent, but it's the manipulation basically of microbes to our benefit. I'm going to shift gears here a little bit, and we're going to take a real quick trip. And you'll see why in just a few minutes, but it's a little deviation from what we've been talking about. 4 years ago, my wife and I had the pleasure of joining another couple, and we traveled to Alaska. And while there, we were able to take a flight seeing tour within Denali National Park. So here we are in a small town called Talkeetna. This is on the outer edge of Denali, which is in south central Alaska. And we booked this flight. They call them flight seeing tours. So you take off from little old Talkeetna, Alaska, and it's about a 20-minute flight into the national park. And we're going to land on a glacier. Yeah, pretty exciting. So these planes are outfitted both with wheels, which you really can't see here, but you can see the skis sticking out. So we use the wheels to take off from the concrete landing pad here, and then we switch to the skis when we get to the glacier. And I was fortunate enough to help co-pilot this de Havilland Otter. That's the name of the plane that we're flying. It's able to take off and land in very, very short distances. So they're great for bush pilots in Alaska due to their ability to not require basically long landing strips. So we took off. We had about a 30-minute flight through some of the most gorgeous country you've ever seen, flying over glaciers. And it was really, really cool. Here we are coming into the Kahiltna Glacier. We're at about 7,000 feet. This is a very long glacier, extends again from the heart of Denali National Forest out into the surrounding area. This is a river of ice, basically, covered here with snow. But just to orient you, here's a base camp. Here on the left-hand side, you can see some of the tents that the climbers stay at for a week or two to get acclimated to the higher elevation. And these structures here that you see are basically colored cones so that we know, the pilot and I know, where to come in and land. Now the snow is a little deceiving because it's hard to get a sense of depth. And these, of course, tell you that we've got a little bit of a hill here before you kind of descend down a little slope. And that can be pretty important to know if you're trying to land or take off on a plane. This is not flat, it's kind of rolling. Denali is the highest peak in North America. So it's over 20,000 feet in elevation, and it is one of the premier sites for mountaineering here in North America, or really around the world. People come from all over the world to climb Denali. There's a rather short time frame that you can climb it. Generally from I'd say June to August is that narrow window that you've got to plan to climb. Unless you want to die, of course, you could climb it now if you want, I suppose, in January, but I wouldn't. So narrow window, and as it mentions here on the slide, this is such a high elevation that you have to contend with high winds, extremely cold temperatures, even in the middle of the summertime. Now, when we landed on the glacier, it was quite nice. It was, people were out in their short sleeves. You know, you gotta be careful you don't get a sunburn due to the reflection of the snow, of the sun off the snow. But as you start to ascend Denali, you're getting into some temperatures that are well below zero, even in the middle of the summertime. So you have to be well prepared for that. And here's my wife Wendy and I in front of our de Havilland otter, and you can see again some of the climbers here at base camp at about 7,000 feet. So they've got 13,000 more feet to go plus to get to the top. So it takes a long time. You stay at several base camps before you can ascend to the summit. And people die every year climbing Denali. It's very treacherous. You better know what you're doing. So you might say, why are we talking about this? What does this have to do with microbiology? Well, other than just being kind of a cool trip, which I hope you all have a chance to visit Denali someday. On our way back from landing there, I caught a glimpse of what looked like blood on the snow. And I took this photograph from the airplane, and it fits right into what we're talking about in this course. What you're seeing here is not blood, but it's actually algae that grows in the snow fields. Yeah, they call it watermelon snow of all things. So again, to give you some idea of how ubiquitous microbes can be, you know, here we are at 7, 8, 10,000 plus feet. You can find this in the lower, contiguous states here of the U.S. as well. Don't have to go to Alaska to see watermelon snow. You can find it in Colorado. I've seen it there before too. But these algae like it cold. And the reason that they're red is due to pigments, as it mentions here, that they have evolved to protect them against the harmful UV rays of the sun. Now, you might remember, I don't know, I hope, that exposure to UV is not good to most cells. We know there's an increased risk of skin cancer if we sit out in the sun a long time. Well, so too can UV be harmful to all types of cells, like the cells here of the algae. So they've evolved the means to protect themselves by producing this red pigmentation. So kind of just an interesting sideline. So I thought you might be interested in hearing about. So as I said a little bit earlier, let's not think of microbes as being always harmful. There are a lot of beneficial ones. A lot of them are very benign. They don't cause us problems. They don't help us. They're sort of commensalistic in that sense. We will be spending quite a bit of time in the course, though, focusing on the pathogenicity of microbes, this notion that some can cause disease, that some can live on or in us and cause us harm, whether that's a cell that's infected with the coronavirus, or it's you getting bitten by a mosquito that transmits the Plasmodium protozoan into your body and you get malaria, or you're injected with the Zika virus, or you may eat food that had an abnormally high number of bacteria in it and you get sick that next day from food poisoning, right? I mean, there's a myriad of examples we can and will talk about as it pertains to the pathogenicity of microbes. So 2,000 plus different kinds of microorganisms that we know can cause disease in humans, more being discovered each and every year. Lots and lots of infections worldwide, 10 billion new infections due to microorganisms. 13 million deaths worldwide, with 90% of those being caused by these 5 major infectious groups. You might remember us talking about these a little bit earlier. So there certainly are a lot of microbes out there that can cause us problems. Lots of good ones too, though. This is just sort of an interesting table that points out some different types of diseases, many of which you've heard of before. Some of these may be new to you. The number of new cases every year, and this is worldwide, and then the number of deaths associated with these different microbial caused diseases. I'm not asking you to memorize this table or anything like that, but I would simply ask you to look through it, get a sense of, the numbers. I think it's kind of interesting. And focus your attention on these two in particular. HIV/AIDS, you've all heard of that. We've certainly come a long way in controlling AIDS, but we still have 1.5 million people dying every year from HIV/AIDS worldwide. That's a significant number of people. And that same number, as you see at the bottom, attributed to this bacterial induced disease called tuberculosis. It's Mycobacterium tuberculosis is the name of the causative agent of this respiratory disease. So we know a lot about pathogenic forms of bacteria and other protozoa and so forth, and they do a job on us, right? As a society, people are dying every day due to microbial activity and infection. This box I just put on here because I thought it was sort of interesting. As of January 23rd, 2021, here in the US, we've had over 400,000 deaths with close to 25 million cases of COVID. Worldwide, almost 100 million cases and over 2 million deaths. And if you go to the Johns Hopkins University COVID-19 dashboard online, which is super interesting, you can kind of get a sense of how prevalent COVID-19 is in the US compared to other countries. And I think you've probably all heard of this, but we're the #1 country in terms of per capita cases and per capita deaths. I mean, we're so #1, there's nobody even close to us. And there's lots of reasons for that, which I won't go into here, but it's certainly worth pondering. This slide kind of talks a little bit about how socioeconomic drivers can influence mortality. So we're looking here at 4 different broad categories of income: the low, low middle, upper middle, and high income countries. Of course, we would be here. Bangladesh would be here, right? Costa Rica might be here. Brazil might be here. And so we're looking at, in blue, the communicable infectious diseases. In yellow, chronic diseases, which we define down here. And green, deaths due to injuries. And not unsurprisingly, or not surprisingly, I should say, not surprisingly, the low-income countries have higher mortality rates across the board, whether it's communicable, chronic, or injuries. With communicable disease transmission sort of falling as we go from low to high income, that's certainly not surprising. Why is that? Well, it boils down to healthcare systems in place to treat the population, right? We don't have many doctors per 100,000 people in certain countries. And so they don't get checked every time they get sick, or they don't have the means to treat the individual if they get sick. So there's higher deaths. But I think what's also quite interesting is the yellow bar here, the chronic diseases. Notice that high income countries have a higher number of mortality, death, due to chronic diseases than do, say, upper middle income countries. And this is no doubt due to our sedentary lifestyle and our great habit of wanting to swing by the fast food, you know, of the local McDonald's or Wendy's or whatever it might be, not to pick on them, because you know, I like an occasional, you know, burger, but we have a mindset, we have an attitude, good or bad, that predisposes us to a greater impact of chronic disease. So anyway, just sort of interesting to think about this in light of what, you know, could perhaps be more controlled if we adopted a different attitude. Well, in some cases, people have no control over a lot of this because of the low income situation. Just interesting to think about. Okay, I'm going to stop here, and on the second half of chapter 1 Zoom presentation, I'll pick up here with historical foundations of microbiology. So get into chapter 1 of your book, start reading it, and we'll stop